Hello everyone. In the previous session we discussed about second model, that is unit membrane model. In this session we discuss another model, that is fluid mosaic model. Fluid mosaic model. This is third model. This fluid mosaic model is proposed by Singer and Nicholson. Who proposed fluid mosaic model? Singer and Nicholson. Singer and Nicholson proposed fluid mosaic model. Singer and Nicholson taken same points which were explained by Dawson and Danieli in the first model. According to Singer and Nicholson, plasma membrane is made up of with proteins and phospholipids. Plasma membrane is made up with proteins and phospholipids. Proteins and phospholipids. This was said by in the first model, that is Dawson and Danieli's model. Okay. Next, proteins. Coming to proteins. Proteins are alpha globular structures. Proteins are alpha globular structures. That means they are spherical structures. This point is said in the first model, Dawson and Danieli. Same points were taken Singer and Nicholson, but here Singer and Nicholson interpretation is different from Dawson and Danieli's interpretation. Okay. Here proteins are alpha globular structures. Again, proteins are two types. They are extrinsic proteins, and second type is intrinsic proteins. Intrinsic proteins. These alpha globular proteins are two types, extrinsic and intrinsic proteins. Coming to extrinsic proteins. Extrinsic proteins are outer side of phospholipids. They are outer side of phospholipids. Outer side of phospholipids. Again, these extrinsic proteins are attached with phosphate group of phospholipids. They are attached with heads of phospholipids. Heads of phospholipids. And extrinsic proteins are loosely attached. Their attachment is loose. Attached loosely. Okay. This is about extrinsic proteins. Coming to intrinsic proteins. Intrinsic proteins are partially or completely embedded in the phospholipid bilayer. They are partially or completely embedded. In phospholipid bilayer. This is about proteins. Are you understand? Proteins are of two types, extrinsic and intrinsic. Extrinsic proteins are present outer side of phospholipids and they are attached with heads of phospholipids and they are loosely attached. Next, coming to intrinsic proteins, they are partially or completely embedded in phospholipid bilayer. Next, coming to phospholipids. Phospholipids, generally phospholipids are amphipathic. amphipathic. What is meant by amphipathic? Already we discussed about amphipathic characters of phospholipids. That means they exhibit two characters, hydrophobic and hydrophilic characters. Phospholipids contain a glycerol head and two fatty acid chains. This glycerol head is attached with phosphate group. This head part is generally hydrophilic. Hydrophilic in nature, and tail part is fatty acid chain, which is hydrophobic in nature. These both characteristics are exhibited by phospholipids. That's why they are called amphipathic molecules or amphipathic substances. Again, these phospholipids exhibit two types of movements. They are transition movement, and second type is flip-flop movement. In the lipid bilayer, phospholipids exhibit two types of movements. They are transition and flip-flop movement. What is transition movement and what is flip-flop movement? Coming to first one, transition movement. Transition movement is nothing but movement of phospholipids. In the same layer. Phospholipids are arranged in two layers, lipid bilayer. In the lipid bilayer, this is one layer and this is another layer. In this, phospholipids are present. Those phospholipids are moving within the same layer. That type of movement is called transition movement. Second type of movement is called flip-flop movement. What is flip-flop movement? In this, movement of phospholipids between lipid bilayer. In this is one layer and this is another layer. In these two layers, phospholipids are moving from this layer to this layer and this layer to this layer. I understand. Within the two layers, phospholipid molecules exchange takes place. That type of movement is called flip-flop movement. Next, Singer and Nicholson explained the structure of plasma membrane. In that, they said plasma membrane is a trilamellar model, trilamellar structure. First, plasma membrane is a trilamellar structure. Trilamellar means three layers are present. In that, middle lipid bilayer is present. Lipid bilayer is present. On the either side of lipid bilayer, proteins are present. Outer protein layer and inner protein layer. Three layers are present in the plasma membrane. That's why it is called trilamellar model. This point is explained in unit membrane model. In the unit membrane model, Robertson said that the plasma membrane is a trilamellar model. Thus, that point also taken in their interpretation. First model and second model, both models are taken into consideration and they interpret their views and explain the structure of fluid mosaic model. Based on these points, let us draw the structure of plasma membrane. That structure of plasma membrane I will explain in the next class. Thank you.